Lee Jones. Lee is the founder, president, and CEO of Rebiotics Incorporated with more than 30 years of experience in the medical technology industry in large and small companies and in academia. Rebiotics, the clinical stage biotechnology company developing a new class of biologic drugs based on live human-derived microbes, was acquired by Ferring Pharmaceuticals in April 2018. Lee has served on several public and private boards and is a member of the Sophia Angel Investment Fund, is an advisor to several small companies. Let's welcome Lee.
Thank you. Is this on right so you can hear me? I appreciate being asked to come here today, but I was just, as I was standing here, there you go. That's probably better. I'll speak into my collar and that's better. I can see a green light. So as I was standing here, I was thinking, there we go. What was worse, to have the lecture right before lunch or after Dr. Kussler? And I think I took the short straw. So let's start there. So my name is Lee Jones, and I'm the president, founder, and CEO of Rebiotics. I am a 1979 chemical engineering graduate from what used to be called the Institute of Technology back then. So I probably am close to like the third oldest person here, so I'm going to claim some credit for that. I have spent my career developing and marketing new products for new customers to meet unmet needs, and particularly unmet medical needs in the medical arena. So I've had over 25 years of experience in medical devices, and I've spent the last 10 in cellular and tissue transplants and microbiome therapeutics. It's been a really interesting career, and I've had a lot of fun. So I want to ask those of you sitting here today, what if you could have the most fun job in the world? You know, if you just picked a time and a place and you said, this is the job that I think would be the most fun, what would it be? Would you be an explorer? Would you save people's lives? Would you be a craftsman that worked on musical instruments? Would you be an architect that built beautiful buildings? Well, I have to tell you, I found my most fun job in a little bit of an unusual way. It was a combination of the University of Minnesota and human poop. And I can honestly say I never expected to be standing here in front of you and talking about this. About 9 years ago, I was CEO in residence at the Office of Technology Commercialization at the University of Minnesota, hanging out while I was looking for my next thing to do. And I ran across a couple of scientists who wanted to patent an idea called fecal transplant. And fecal transplant is where you take fecal material from healthy people and you put it into sick people to cure their disease. And I was sitting there and I thought, you know, that's the stupidest, most ridiculous idea I've ever heard. I want to help out with that. So I immediately volunteered, because I was really curious about what could I be, to help them look at, could this be a potential business opportunity? Could it be a licensing opportunity, et cetera? And unfortunately, these particular scientists took money from a different group and then they went on their merry way. But I found that I was just captivated by the whole concept. So in 2011, I went off on my own, found my own scientists and money, and I started a company called Rebiotics. So our mission at Rebiotics is to revolutionize the treatment of debilitating diseases by harnessing the power of the human gut microbiome to greatly improve lives. We actually harvest today organisms out of human fecal material, preserve them, package them, and deliver them to patients who are sick. So how did I get here? You think back, it's not something you really typically brag about to your kids. We were talking about dinner conversations, or breakfast conversations. So I graduated, like I said, in 1979 with a chemical engineering degree from the university. And my last year, my senior year, I actually took a course from Dr. Keller on biomedical engineering. He had brought in some samples of heart valves, and my imagination got captured. I thought, you know, I can design things that can help save people's lives. And it influenced me enough that 40 years later, I'm still doing the same thing. I've had a number of different positions. I've been a CEO of other companies. I've worked here at the university. I ran the Diabetes Institute and then founded Rebiotics. So what is the microbiome and why is it so important? Well, the microbiome, if you've seen anything lately, if you even watch television, you can hear about the microbiome. I was at home, I had the TV turned on, and I saw an ad from Dove Body Wash, and it said, "Does your body wash influence your microbiome?" And that's when I knew we had gone mainstream. The microbiome, all of our surfaces are covered in trillions of microorganisms. And these microorganisms work together symbiotically to help us stay healthy. The most diverse and dense microbial community in the human body is in the human intestinal tract. And what's important about that is that that community works also together to keep us healthy. But when it gets disrupted or it becomes what we call dysbiotic through the use of antibiotics or something else, it can leave us susceptible to disease. In fact, research has shown that the gut microbiome is linked to a whole variety of diseases. Gastrointestinal diseases, liver diseases, diabetes, and a number of other things. Particularly what I find most interesting, and there's been research that's been published recently, the gut microbiome is linked to autism and Parkinson's diseases, diseases that we traditionally think of as brain diseases, but maybe they're really not. Maybe we've been treating the wrong organ for a long time. So the question was, if dysbiosis can lead to disease, if we fix that dysbiosis and put people back into a normal state, can we then cure the disease? So that's a question that we set out to answer. We knew from past history that people were using fecal transplants to treat Clostridium difficile infection, which has been designated as an urgent national threat by the CDC. People who get C. diff typically have a damaged microbiome. It's a horrible gastrointestinal disease that is infectious and can be transmitted to other people. And it's characterized by severe diarrhea and really, in some cases, it can be deadly. So we thought if we could take our product and treat these patients, could we affect that? Because what happens with these people is they take antibiotics for some reason. Their microbiome is damaged, they get exposed to C. diff, they get C. diff, and what do you think they do? They take more antibiotics to treat the C. diff. And then what happens again? They get the disease again. So there's a number of people that end up in this spiral, almost of despair. They're desperate because they can't kick the disease and they can't find anything to treat it. So that was our first mission. So I went to the FDA in 2012 and said, hey, we've got this great idea. We're taking microbes from human stool and we're putting it in other humans. It should be a tissue transplant, just like a heart transplant. You take heart out and put it in somebody else. And the FDA said, good argument, but is that the microbes you're dealing with are not human. So you are developing a new biologic drug and you are being regulated in the Center for Biologics Evaluation and Research, CBER, in the vaccines division. Now, I came out of the medical device industry. I knew nothing at all about drug development. And not only did I not know anything about drug development, but there was no regulatory pathway for this new type of drug. We were the first company to take this type of product through the FDA, and we faced a number of challenges as a result of that. There was no regulatory precedent, so we couldn't look at some other company or somebody else and say, well, that's how they did it, that's how we should do it. There was no manufacturing guidance, no quality guidance, no way to test anything, and really a lot of hype, but no real clinical data. So everybody believed this would work, but there was really no proof. So it was hard to even know where to get started. So what we decided to do was find some donors, get our hands on some raw material, and start experimenting. And it's been interesting. So I thought, well, this is poop. People have been doing this for a long time. Really, how hard could this be? Really, how hard could this be? And when I found out, it was totally different than I expected. We had no way to determine what was in our mix. We didn't know how to test anything. we didn't even know, what a dose could be. So if you think about most drugs, like an aspirin or something, you take it a couple hours later, it goes away, and you take it again. Well, in our case, we give the patients a dose of our product. It's alive, so it starts to grow and multiply. So you start thinking, okay, what does this mean? Is this a dose? And how does that work? So what our goal was, through the course of our product development, was to keep as many microbes and as diverse a mix of microbes alive in our product as we could. And what we found was that there was some really serious science required in order to have this happen. Now, these organisms are anaerobes, which means they don't live in air. So you have to process them and keep them in a non-oxygen environment. So we started conducting multitudes of experiments. I think we ran thousands of experiments in experimental designs. We mapped the output parameters, and we had to develop our own test methods, so we knew if we were making a difference when we were doing things on our processes. And we eventually came out and discovered and created, I should say, our microbiota restoration drug product platform. Now, in our product platform today consists of 2 drugs, RBX 2660, which is a broad spectrum liquid microbial suspension that we deliver via enema, and RBX 7455, which is the first of its kind oral form that's room temperature stable. But both of these products mimic a normal, healthy human gut microbiome. For those of you that aren't in the drug development business like I wasn't, I didn't know that there was vast numbers of clinical trials that had to run to make sure that your product worked and it was safe. And this is sort of how it goes. You start out everything, maybe you have animal studies and you work your way through, and if it all goes well at the end, we were talking about duds earlier and how there can be a lot of duds, but if it all goes through, you get FDA approval. So we've developed a robust clinical program. We are currently on our 4th clinical study, which is a phase 3 study, so our last one that we think before we can go to marketing. And we are the most clinically successful company in the microbiome space worldwide. We truly believe that we'll be the first ones ever to have a product like this on the market. Well, clinical studies come with a price tag and a lot of time involved. And we were a small company, so we're getting investment dollars to do this. We had to really design these well. A typical clinical study for us would look like this. The patients we would see would typically have 2 or more episodes of this disease before they'd be eligible for our trial. Typically they were on antibiotics, so we have to take them off the antibiotic regimen they were on. We would test them at baseline, and then give them our product, and then follow them and see what happened. In our world, 8 weeks. If they can go 8 weeks without getting the disease again, we consider that a success. In the meantime, we were taking stool samples all along the way, because our goal was to start looking, how is that microbiome changing as their disease goes away or progresses, we'll see. And what we found is that our microbiota restoration therapy had a significant impact on dysbiosis. What you see in this chart here is what we call a relative abundance chart, the spider web diagram from the bottom. You can see it on the left-hand side what our product looked like. There's 4 main classes of organisms. We had a high abundance of Bacteroideia and Clostridia. The middle one is the patients, what their microbiomes looked like with those 4 classes when they were ill. You can see the totally different high levels of pathogenic bacteria. And then 60 days after being treated, their microbiomes reverted to look more like a normal, healthy person. Another way that we can illustrate this is what we call this multidimensional similarity analysis. Each one of these dots is a 2-dimensional representation of an entire microbiome for somebody. The black dots on the left are from healthy humans from the Human Microbiome Project. The dots on the right, the white ones, are our patients at baseline before treated, and that little triangle is kind of the average of that patient group. So this is at baseline before treatment, 7 days post-treatment, 30 days post-treatment, and 60 days post-treatment. So after treatment, the patient's microbiomes progressively shifted closer to the normal human microbiome profile. So what we found is that our RBX 2660 breaks that cycle of C. diff infection. So remember I told you if people take antibiotics, they end up with a disrupted microbiome. They can get exposed to C. diff, they can get the disease. But by giving them RBX 2660, it actually eliminates the need for those antibiotic treatments. So why is this important? Well, this is proof of concept here. What we've shown is that we can, by putting the microbiome back to a normal healthy state, that person fights off their own disease. We're not killing organisms, we're not like an antibiotic, but we're actually helping somebody to treat themselves as time goes on. And what this does is opens up a whole new world of research for us. So if you take that same principle and you apply it to a number of other diseases, you might have almost what we call miracle cures, or really change the way that person's life could progress. So we have clinical studies undergoing today in recurrent urinary tract infections, pediatric ulcerative colitis, and a number of other things. So I think it's really exciting, and I'm certainly an evangelist for this. So remember I asked you what, if you had the most fun job in the world, what would it be? Well, I have to tell you, I found mine, and I would say that the CEMS is a good place to start. So thank you.